Good morning. Today we are going to deal with the most interesting portion of the nervous system and one of its functions that will take the second hour. So we'll start with the hypothalamus and the main things, and then we are going to continue with the autonomic nervous system. So the general objectives then of these 2 hours will be to understand the significance of the hypothalamus, the general organization of the hypothalamus, and examples of its functional and dysfunctional states. After that, we'll go into the autonomic nervous system, that we'll see how is the general organization of the visceral innervation, and then select a few subjects for going deeper into the matter, and we'll go into the neural control of the blood pressure, of the bladder, urinary bladder, and of sexual function. To start then with the general significance of the hypothalamus, is the most impressive, extraordinary piece of nervous system. Why? It only weighs about 4 grams in the adult brain, and look at what 4 grams of brain tissue can do. 4 grams means something like 0.05% of the total mass. Is minimal. And look what all that can do. It regulates homeostasis, and it regulates the body temperature and hunger and thirst and emotional states and circadian rhythms and sleep and wakefulness and reproductive function. There is very little remaining after all these controls, and it does that through the control of the endocrine system and the autonomic nervous system. So we will go now to review the general organization of the system, and then we'll end up with the autonomic nervous system. We'll leave the endocrine system aside, since you get a full course on it separately. Now, the general organization of the hypothalamus, we have to have some idea where it's located and in various portions, because it affects differentially its functions. Now, I'm presented here some sections that are actually from monkey brain. They are very, very similar to the human, almost identical, I would say, and they are a little reduced in size. And I do that because the hypothalamus is a particularly difficult area to fix, to get fixation of the human brain, and to obtain sections of it is quite difficult. It's not impossible, it's quite difficult. So I'm trying to bypass that difficulty by showing sections of a monkey brain. Here you have a mid-sagittal section, and you can recognize, I'm sure, the corpus callosum, and you can recognize here the fornix, and that would be the anterior commissure, and that's the optic chiasm. And then you have all this region that I outlined here, that is the hypothalamus. And on top of that, it will be the thalamus, right? That's posterior commissure, this is the pineal gland. So you have some general orientation. So you see that is a very small piece of tissue, but quite well organized and in differential zones. Let's take a look at that a moment. This, what I indicated here, is the pituitary gland that is hanging from the hypothalamus, contained within the sella turcica. Now, let's cut this brain in coronal sections and see how the hypothalamus looks from rostral to caudal. First of all, you have a section indicated by this line that I put here, all right? And that's this section. You can it is in front actually of the optic pathways, the optic tract and the posterior chiasma, and therefore this is called the preoptic region of the hypothalamus. You can recognize here again the anterior commissure and here the optic chiasm, and all this tissue, great tissue that is here, is the preoptic region of the hypothalamus. You can see as an overview that you don't see discrete nuclear masses here, but it's quite a number of neurons more or less spread out in this general area. That general area, this is the structure that I mentioned to you before, that general area is what is called the preoptic region of the hypothalamus with no particular nuclei. However, if you look carefully at the dorsal surface of the chiasm when it touches the hypothalamus here, there is a very tiny nucleus on each side of the midline that is called the suprachiasmatic nucleus that I indicated by these letters here. The suprachiasmatic nucleus receives direct input from fibers in the optic chiasm and is the internal biologic clock that varies its frequency of discharge and its activity according to more or less a diurnal pattern of 24 plus hours. And we'll discuss that eventually. All right, so this is the preoptic region of the hypothalamus. Now we make another cut like this one here, a little more posterior, all right? And that is an area that is just on top of the visual pathways. These are the two optic tracts, right? This and this are the two optic tracts. This is the pituitary stalk hanging from it. And that's this region of the hypothalamus that is on top of the optic tracts, is called the supraoptic region of the hypothalamus. And here you can see some discrete nuclear masses. You see one here and you see one here. One near the wall, this is the third ventricle, so this is the wall of the ventricle, and that's what is called the paraventricular nucleus, and this here will be the supraoptic nucleus. And finally, the rest of the region, again, is a more or less amorphous spread of neurons that actually blends with the preoptic. You see, in the nervous system, you don't have a delineated cut that is one side of the cut is something else, and the other side of the cut will be even another thing. Here is like a blend of this portion of the supraoptic region with the preoptic. And that portion of the supraoptic region is called the anterior hypothalamus. So usually the region is referred with the two words. This is the preoptic anterior hypothalamus is one entity functionally. Now we do a third cut, as I showed here. And this third cut, you see, is what is called the tuberal region of the hypothalamus. You have the two optic tracts here that are separated already. They are diverging, going towards the geniculate, as we discussed earlier. And then between the two optic tracts, you have all this gray mass of the hypothalamus that you can see very well if you examine the brain at the basal surface. And it's like a gray blob there of tissue. And that's what is called the tuber cinereum, and that's why this region is called the tuberal region. It contains many nuclei, many subnuclei. But one main subdivision that occurs in the tuberal region is being given by the passage of the column of the fornix. Remember, this fornix bends down and is split by the anterior commissure and goes through the tuberal region to eventually end in the next region, which contains the mammillary bodies. Right? So in going through this region, it actually splits it into two: a medial region and a lateral region. And finally, you have a nucleus that actually goes from one side to the other, closing the ventricle here on top of the pituitary stalk, that is called the arcuate nucleus. So you have the medial hypothalamus, the lateral hypothalamus, and the arcuate nucleus. Of course, both the medial and the lateral hypothalamus have several subnuclei on it that we may refer to them functionally. Finally, if you make another cut like this, all right, you go into the so-called mammillary region, and the mammillary region contains the mammillary bodies, which in the human, they're a little more prominent in the subarachnoid space. They are less prominent here in the monkey. All right, this is the region. Here you have the thalamus, for instance, and here you have the subthalamic nucleus, and here you have the substantia nigra. Right, and here is the mammillary bodies. Now, the mammillary bodies have a more anterior region to the actual bodies, all right, that actually blends with the posterior hypothalamus, which is part of the lateral hypothalamus. All right, it blends again, the same that was happening with the anterior hypothalamus and the preoptic region. And this is a very important nuclear mass that is called the tuberomammillary nucleus, between the tuberal region and the mammillary region. It's important because here is a group that manufacture histamine and provide histaminergic innervation to the thalamus and the rest of the brain. Actually, is the only nucleus known that contains these histaminergic neurons. And as we are going to see, they are important for alertness, whatever, you will see it in a moment. All right, so this is the basic layout of the hypothalamus. You have a preoptic, a supraoptic, a tuberal, and a mammillary region. Now, we are going to go into some connections of these various nuclei. We'll take it as a whole, the whole hypothalamus. It receives inputs and provides outputs. And of course, what happens within the hypothalamus, which is the intrinsic organization, is extremely complex, and we'll touch on it in a moment. But let's go to some relatively discrete inputs. All right, the hypothalamus receives a very important limbic input. Right? In the limbic system that we didn't have opportunity to discuss, there are two major structures that provide outputs of the system. One is the hippocampus fimbria fornix system, and the other is the amygdala. The hippocampus fimbria fornix system, eventually, as we discussed other times, the fornix will bend into the column of the fornix, which I said before, meets the anterior commissure, gets split, and the post-commissural portion, which is the major portion of the fornix, is going to pierce through the tuberal region and go to the mammillary region, where it terminates in the mammillary bodies. They are very concerned with memory processes, as you may know already from studying the limbic system. Now, the other input from the limbic system comes from the amygdala. The amygdala gives off an important bundle of fibers, which is the stria terminalis, that is a C-shaped structure that goes between the caudate and the thalamus, and then eventually bends and pierces through again and goes to terminate in the preoptic anterior hypothalamus. As you well know, the amygdala has been very much related to the emotional content of memories, and so you have now that the preoptic anterior hypothalamus is part of that same system. Now, in addition to input from the limbic system, the hypothalamus also receives input from almost all the sensory systems. First of all, receive visual input, that I just mentioned, right? These fibers that are coming off the chiasm and terminate in the suprachiasmatic nucleus, which is responsible for circadian rhythms that we'll discuss in a moment a little further. Then it receives an important input from olfaction and taste. And these inputs from olfaction, for instance, is coming actually from a portion of the amygdala, from the corticomedial amygdala. And both the smell and taste, which is the chemical, so-called chemical senses, has these attributes that not only have a discriminative aspect that allows you to identify a particular odor or a particular taste, that's done at the cortical level, but I have also these motivational aspects in terms of qualifying your stimulus as pleasant or unpleasant, all right? And this is carried on most probably through networks in the preoptic anterior hypothalamus that receive these additional inputs from smell and taste. Then you have also somatic input, somatosensory input, that is very clear from erogenous areas, and is also from pain, from the pain system. There is direct input into tuberal regions of the hypothalamus. Then you have input from the visceral sensory systems, right? And most regions of the hypothalamus receive input that is carried from projections from the solitary nucleus. The nucleus of the salivary tract, as you may remember, receives the fibers from several cranial nerves, particularly from the vagus, that are carrying information of sensory inputs from the viscera. And eventually, the solitary nucleus, one of the projections, it projects to many places, one of the projections is actually the hypothalamus. Most regions receive that. Finally, there is good evidence that the vestibular system also carries an input into the hypothalamus, particularly into the supraoptic nucleus, that the supraoptic nucleus fabricates vasopressin. And it is known that one of the earliest controls of blood pressure when you go from a reclining to a stand-up position that requires an adjustment of the blood pressure because the mass of the blood goes essentially when you stand up to your legs and to your abdomen, and you need blood upstairs. And so there are adjustments that are made, and one of the adjustments is carried by the vestibular system that samples, as you may remember, the position of the body in space, and that results in a surge of vasopressin, that one of which functions is vasoconstriction. It's also known that in motion sickness, one of the first things that occur is a surge of vasopressin. Finally, the hypothalamus has a different kind of input that is not neural, is humoral. There are several portions of the hypothalamus that have very leaky brain-blood barrier, and therefore is very much influenced by certain components of blood, such as glucose levels, for instance, and also various hormones of all kinds, and finally also for a substance that is secreted by adipocytes that is called leptin, that in turn controls to some extent feeding functions, as we are going to see. So these are essentially the main inputs to the hypothalamus: limbic, all sensory, and humoral. We go now to the outputs of the hypothalamus. First of all, there is a very important control coming out of the hypothalamus to modulate the autonomic nervous system. And it's known that the anterior and medial hypothalamus have to do a lot with parasympathetic control. These are essentially the origin of the preganglionic parasympathetic fibers that are going to terminate either in cranial nerve nuclei or in the sacral cord. On the other hand, in general, the lateral and posterior hypothalamus have a lot to do with sympathetic control, which means that these are preganglionic fibers that come down to the thoracolumbar spinal cord to activate the nuclei in the lateral horns of the spinal cord that will give off the sympathetic outflow. Another important output of the hypothalamus that I I'm not going to discuss in detail because you're going, first of all, I'm sure you know it already, and second, it would be very much discussed in the endocrine course, which is the hypothalamic hypophyseal tract that is called, hypophyseal being the pituitary, all right, that is coming essentially from axons of the supraoptic and paraventricular nuclei, the magnocellular portion of the paraventricular nuclei. These are neurons in these nuclei. The release of their own substance that they fabricate, that in this case will be vasopressin and oxytocin, are going to actually come out of the nerve terminals in the standard capillary bed of the pituitary stalk and even of the posterior pituitary, and are going to go directly into the bloodstream. So these are outputs of the hypothalamus that go to hypophyseal capillaries for the release of vasopressin and oxytocin in the general circulation. Then you have the tuberomammillary nucleus, as I mentioned before, fabricates histamine and provides this innervation to the cortex and the thalamus and other regions of the nervous system. Then from the mammillary bodies, you have a very discrete tract that is the mammillothalamic tract that is going to terminate in a group of nuclei in the thalamus that has a lot to do with the limbic system. These are the anterior group of thalamic nuclei that are part of Pape's circuit, one of the limbic circuits that you must have studied already. And lastly, you have the mammillotegmental tract that comes directly from mammillary bodies into the midbrain tegmentum and carries descending influences. What I'm missing here, that again, I'm missing it because I don't want to go into that subject, you have a full course on it, is the fact that there are several, if not many, nuclear groups in the hypothalamus that manufacture different peptides. And these peptides, now when the neurons are excited, they are released into the portal capillary bed of the hypothalamus and carried through the bloodstream to the anterior pituitary to either stimulate or inhibit the release of hormones of all kinds in the anterior pituitary. These are what are called the various hormone-releasing hormones, or actually, like for instance, the gonadotropin hormone-releasing hormone, and the thyrotrophic-releasing hormone, and that they are all stimulating the secretion of these hormones by the anterior pituitary. But you have also inhibiting so-called releasing hormones from the hypothalamus. For instance, a group manufactures somatostatin that is carried to the anterior pituitary, and what's going to happen there is going to inhibit the release of growth hormone. And even more, the hypothalamus has also a group of dopaminergic neurons in the arcuate nucleus that the release of dopamine into the portal capillary bed is going to carry the dopamine to the anterior pituitary, and there is going to inhibit the release of prolactin. So all of these are, of course, endocrinological details that you will have in the corresponding course in detail, but I didn't want to miss mentioning it. All right, so we have then an idea of inputs and outputs of the hypothalamus. Now let's go a little bit into the intrinsic circuitry, and please do not be hung up on it because it's complicated, but I want to show you examples of the complexity of the intrinsic circuitry with these 4 grams of brain tissue. So there is very complex, as I mentioned here, interactions between specific cell groups. And in example, there are circuits, intrinsic circuits within the hypothalamus that had to do, for instance, with sleep and wakefulness, others that are related to food intake, others that are related to circadian control, others that act as thermal regulators, and between these discrete circuits, there are interactions because you know that usually the animals, humans sleep at night, and they don't feed at night. They feed during the day. So obviously you have already an interaction between the circadian rhythm and the food intake. And also with sleep, because you sleep at night and not during the day, normally, right? And so forth and so on. So let's take a sample, but again, please do not try to memorize it by no means, and have it just as an example of intrinsic circuitry within the hypothalamus to control these functions. So at this point, we are ignoring the extrinsic connections that we dealt with before, right? The inputs, limbic and sensory, and the outputs, all the various systems that I mentioned. Now we deal just purely with intrinsic connections. And look what happens. Here you have a list of abbreviations, all right, of various nuclei and nuclear groups that we are going to deal with. All right, for instance, and here in the color key, you have the various systems that we are going to discuss. So let's start with the sleep regulation. You have here a nuclear group in what is called the lateral preoptic region. Lateral preoptic region. These are neurons that are GABAergic, and therefore they are inhibitory in this nucleus. And this is the main nucleus where something X is acting upon to induce sleep. What is this substance is not 100% known, but this is something that accumulates during the waking state. And when it reaches a certain level, it activates this nucleus that is important for falling asleep, the lateral preoptic. But here you have another nucleus in the hypothalamus that we reviewed before, the tuberomammillary nucleus that manufactures histamine and has also a GABA. I don't know precisely whether this is co-localized or there are different groups, but what is definite is that there is a reciprocal inhibition between these two nuclei. And that's a very peculiar arrangement, two mutually inhibitory structures that remind in electronics something about the bimodal flip-flop switches. Now, you have another thing in the hypothalamus, in the lateral hypothalamus now, that is a group of cells that secrete hypocretin. Hypocretin or orexin is also called, right? That has an excitatory action on the tuberomammillary nucleus, and that they have to do a lot with awakening, to keep you awake. Now, before we proceed further, know the following, that the lateral preoptic region was important for falling asleep is actually decreasing in cell numbers with age, and could be this factor the responsible for sleep disturbances in the aged, that people have problems in falling asleep and have problems in fragmentation of sleep, that you wake up to go to the bathroom or whatever, and then it's difficult to fall asleep again. And that all of this could be due to this observed decrease in the number of neurons in the lateral preoptic nucleus here. On the other hand, you have here histamine that is, as you can see, is inhibiting this previous nucleus under certain condition, all right, and therefore is keeping you awake. And that's, what you have to have clear in mind, that the action of antihistaminics that you take many times, makes you fall asleep, makes you very sleepy, all right? And that's because the histamine is very important in the activation of structures like the thalamus and cortex, and at the same time, it inhibits the nucleus that is responsible for you actually falling asleep. And here in the hypocretin nucleus here that excites the tuberomammillary, it is known that people that have these episodes of sudden sleepiness and during the day, a condition that is called narcolepsy, in narcolepsy that sometimes even results in cataplexia, which is to the falling down asleep on the floor. These people have a very important deficit in this nucleus that secretes hypocretin, right? So you have just intrinsically within the thalamus a whole circuitry here that I'm simplifying to the bare minimum that has to do with the control of sleep and wakefulness. Now let's do something about feeding, feeding circuits. All right, you have in the blood circulating substances. One of them is leptin, that I said before, that is produced by adipocytes, right? So this leptin has an action on some nuclear masses in the hypothalamus directly, right? You have that in the ventromedial hypothalamus and the arcuate nucleus, there are groups of cells that are so-called orexigenic, which means that they promote appetite, and other groups that are called anorexigenics, just the reverse, all right? And there is a interaction between these two groups so that the orexigenic are inhibiting here, they are GABAergic, and they inhibit the anorexigenic. Now, take the case of what is happening with leptin. Well, when you have a lot of leptin in the blood, because your adipocytes are secreting that substance, all right? The leptin reaching the hypothalamus is going to stimulate the anorexigenic and are going to inhibit the orexigenic. All right, if you take one thing at a time, if they excite the anorexigenic, this group of cells in the ventromedial hypothalamus is going to act on the paraventricular nucleus of the hypothalamus also, the parvocellular portion, all right, through the production of pro-opiomelanocortin and alpha melanocyte stimulating hormone. These two products of the anorexigenic group are going to activate the paraventricular nucleus of the hypothalamus in a particular fashion that results in the suppression of appetite. So when you have a lot of leptin in blood that is exciting this anorexigenic group, you get a suppression of appetite, so you don't eat so much. At the same time, the leptin is inhibit the orexigenic group. And when there is a lot of leptin, this group is inhibited. But when the leptin goes down, when the adipocytes do not secrete so much leptin, you will see that the orexigenic group, there is a production by these cell groups of neuropeptide Y that in turn will act on the paraventricular nucleus of the hypothalamus and result in appetite stimulation. So when you have low leptin, you will have increased appetite. All right, so you have one circuit for sleep, one circuit for intake of food. Let's do one more circuit for circadian rhythms. You have this suprachiasmatic nucleus that receives input from the retina and that is entrained by the diurnal cycle. So it discharges maximally in daylight, in full daylight. And it acts through intermediate networks. Here I'm really simplifying, but through intermediate networks is such that it stimulated the dorsomedial hypothalamus, another region of the hypothalamus. It's stimulating that, and the dorsomedial hypothalamus in turn is inhibiting the lateral preoptic. Well, during the daytime, when you have a lot of suprachiasmatic activity and a lot of dorsomedial thalamus activity, the nucleus that is responsible for sleep is inhibited, and you expect that. You don't sleep during the day, right? In addition, you have that all this circuitry for feeding is also exciting the dorsomedial nucleus, all right? And so by giving you the possibility of eating in your wakeful state. And in turn, the dorsomedial also has an excitatory action on the hypocretin, again awakening you. So you have this interaction between the suprachiasmatic, the circadian rhythm circle, and both the sleep circle circuit and the feeding circuit. There is also an action of the suprachiasmatic diurnal pattern on the medial preoptic that has to do mostly with control of body temperature. Now, what I'm not, I think that we'll discuss sleep. We'll go a little more into this circuitry, all right? The role of the pineal gland, the role of melatonin, but this will leave for the lecture on sleep mechanisms. All right, so here you have now an example of intrinsic connections within this tiny bit of brain. Now, functional and dysfunctional states. What's going to happen here? Well, I have to talk in terms of very general terms, all right? And I mentioned to some extent this. When there is activation of the anterior or medial hypothalamus, that results in a parasympathetic effect. Now, what's the parasympathetic effect? Well, the pupil will constrict, the heart will slow down, the gut will constrict more, all parasympathetic effects, all right? Whereas activation of the lateral and posterior hypothalamus has just the reverse, has sympathetic effects. The pupil will dilate, the heart will increase its rate, the gut will slow down. All right, that's in very, very general terms. Now, examples of that in the sleep-wakefulness cycle, I did mention it when we discussed the intrinsic connections, that you have these hypocretin secreting neurons in the lateral hypothalamus that excite tuberomammillary nucleus, histaminergic, is a review actually, which in turn activates the thalamus and brain structures. Right? And the loss of these cells can lead to characteristic disease states like narcolepsy. On the other hand, decrease in the neurons of the lateral preoptic or in the aging process can be responsible for the problems in sleep that old people have, like me. Anyway, temperature regulation. This is very characteristic in terms that the anterior hypothalamus contains a group of cells that are very sensitive to increases in the temperature of the blood. When the temperature of the blood increases, it elicits compensatory mechanisms that result in a heat dissipation. To increase temperature, you have to dissipate heat, right? Heat dissipation, mechanisms like vasodilation. You become red and you release more heat, all right? And you also sweat. And that occurs what is characteristic when this group of cells is inhibited, and it is inhibited particularly for degradation substances of bacteria, when you have an infection, but also prostaglandin E2. What is going to happen when this group is inhibited? Your temperature will rise. You have the result is fever. All right? Hyperthermia or fever is the result of inhibition of this group of cells that are reactive to increase heat in the blood. Similarly, there are now in the posterior hypothalamus neurons that respond to blood cooling. And when blood cools, the mechanism to compensate for that were mechanisms to retain heat as vasoconstriction and to generate heat like shivering, right? To compensate for the low temperature in the blood. When you have problems with this region, remember problems with the other region was giving you fever. Here, it doesn't give you just low temperature. The person or the animal, experimental animal, becomes totally non-regulated for temperature, and that's what happened in cold-blooded animals, right? If you put the person in a cold environment, the temperature will go lower. If it's the hot environment, the temperature was higher. And that's what is called poikilothermia here. Why this is happening? I think it's because the output of the anterior hypothalamus, all right, got the fibers carrying all these functions, mostly parasympathetic, are going through the posterior hypothalamus to go to brainstem regions. And therefore, when you have a lesion in the posterior hypothalamus, it also includes a problem of the temperature regulation for heat. All right, so that for that. Now, another thing, another functional and dysfunctional state occurs with food intake regulation. And you know, the tuberal region, you know, the response to visual sensation, visceral sensations, and also to the olfactory and limbic system input, and the blood levels of glucose, of hormones, leptins, and so forth. And it acts, this tuberal region, either these factors that are coming into the hypothalamus are activating either hunger or feeling or feeding mechanisms, all right, by activating orexogenic groups, all right, and lesions may result in anorexia. It's not very clear, but it's possible. And also there are just the reverse of hunger, which are satiety mechanisms in the medial hypothalamus, all right, the lesions of which will result in obesity, in increased food intake, obesity, and usually goes together with some secondary sexual features, and they result therefore in some kind of retarded sexual development, and as is characteristic of the so-called adipose genital syndrome or dystrophy. All right, take-home messages of this. The hypothalamus is a very, very, very important piece of brain. It receives information from the limbic system and the sensory systems. It controls the autonomic nervous system and the endocrine system. And it regulates all kinds of functions: body temperature, homeostasis, food and water intake, emotional states, circadian rhythms, sleep wakefulness, reproductive function, the whole thing. The organization anatomically is in 4 regions, from rostral to caudal, preoptic, supraoptic, tuberal, and mammillary, and with several nuclear groups in each of the regions. And there are signs of dysfunction. In the signs of dysfunction in the preoptic anterior hypothalamus can be sleep disturbances, can be hyperthermia or fever, and problems in the supraoptic region. You can get diabetes insipidus in terms that the hypothalamus doesn't produce enough vasopressin. The medial tuberal region, reproductive disturbances, obesity. And the lateral region, you have poikilothermia and anorexia. In the lateral hypothalamus, you can have narcolepsia. In the mammillary nucleus, you have dementia, in terms of alterations in Papes circuit that give rise to these demented forms like Korsakoff's, and that we have discussed later on in thalamus. All right. So now, if the time allows, we'll continue with the second part of this segment, which is the autonomic nervous system. Now, which is really manipulated and controlled by the hypothalamus in the major form, as we have seen. Now, the first thing to have in mind is, I said, wait, is it really autonomic? Why is it called autonomic? It was called autonomic because it was a system that was not in the central nervous system. It was really peripheral and functioned by itself without the nervous system. This is not the case, all right? So it's a designation that has been adopted and will keep, but know that there is nothing autonomic about the autonomic nervous system, except perhaps in the innervation of the gut as a very unique feature. So, you know that the afferents from the viscera are coming to the central nervous system, right? These are general visceral afferent fibers. You know also that the preganglionic fibers, general visceral efferents, originate in the central nervous system. And also have in mind that the autonomic ganglia, once outside the central nervous system, contain networks. They are not simply relay stations. Now, this is an old cartoon from the development of spinal cord that eventually gives rise to this 4 kinds of fibers in the spinal nerve, all right? But now let's consider the autonomic ganglion. In the autonomic ganglion, you have an input to the autonomic ganglion that is coming, let's say, from these general visceral efferent fibers, all right? They come into the autonomic ganglia, they output the spinal cord to what is called the white ramus. And now the neuroactive substance released by these terminals of these neurons in the central nervous system is essentially acetylcholine. Now, the acetylcholine is going to activate neurons in the autonomic ganglion, the axons of which will leave the ganglion. These are projection neurons. They leave the ganglion, they go into the great ramus, and they distribute in the peripheral nerve, right? And the neuroactive substance that they release is norepinephrine. All right, that will be just a simple relay. But the point is that the autonomic ganglion has other cell types in addition to the projection neurons, and one of these, there are several, one of these, for instance, is a neuron that is dopaminergic, right? And that these neurons are called SIF cells from small immunofluorescent neurons. Never mind, they are monoaminergic. All right, and this particular one is dopaminergic, and you see that it's activated by the same cholinergic input here. So you have the different actions of these various synapses in terms of the receptors that are impinged. For instance, here the synapse between the incoming cholinergic fiber and the input of the cholinergic fiber onto the projection fiber is a nicotinic receptor, whereas the input of the cholinergic fiber on the SIF cells is a muscarinic receptor. And finally, of course, here it will be in the receptors, in the projection neuron, it will be a dopamine receptor. So there are all kinds of interactions, there are plenty of neuropeptides within the ganglion, and there is a lot of processing within the ganglion, and eventually the result of that processing will come to the periphery. So have this in mind, because I think it's quite important. Because it's important because most of the manipulations that you do therapeutically to act on the system is essentially addressing these other connectivities within the autonomic ganglion and not the direct output, because the direct output, the only thing that you can do is to eliminate it. You cannot modify it per se. All right, now we go into continue with some general points on the system, all right? For instance, at the brainstem level in the upper medulla, there is a cartoon that shows here the ample fourth ventricle, and this is the upper medulla, so you have nuclear groups on the floor of the ventricle. The midline is here. This is the terminal sulcus. The midline is here, all right? So you have motor groups near the midline, and then until you get the sulcus limitans, and then you start getting, and close to it you have visceral nuclei, and then you have visceral sensory nuclei, and eventually somatic sensory nuclei. All right. Now, in terms of the location of nuclei, you have this little table. This is the midline, all right? And you have here various columns. The close to the midline is general somatic efferents, then it will be general visceral efferents already here, and then general visceral afferents. And we are interested in the visceral efferent and afferents. All right, here you have the levels of the brainstem. All right, so you have the midbrain, and you have the pons, and you have the medulla here. All right, this happens to be the medulla level. All right. Then you have your first column of general somatic efferents. We are not interested. We are not dealing with that at this point, all right? But know that they go through if you review cranial nerves, you have nuclei of the third and the fourth nerve, and at the pons level, the 6th, and the medulla level, the 12th. That's all right. These are all motor. But now when you go to the general visceral efferent columns, you have the motor nuclei of the visceral system. That you have at the level of the midbrain, you have what is called the Edinger-Westphal nucleus that you may remember from the lectures on the visual system. All right, these are essentially the preganglionic parasympathetic fibers of the third nerve that will take care of the constriction of the pupil. Then at the pons level, you have at the caudal pons level, you have the superior salivatory nucleus. The fibers are being carried by the seventh nerve. And at the rostral medullar level, you have the inferior salivatory nuclei that the fibers are carried by the ninth nerve. These know their existence. You cannot really see them in the sections because they are not discrete nuclear groupings. They are more scattered cells that have these functions. Finally, at the medulla level, you have a most important nucleus that is the dorsal motor nucleus of the 10th nerve that is represented here. The dorsal motor nucleus of the third nerve, all right? These are preganglionic parasympathetic fibers, all right, that they come to the internal organs, in this case, the intramural plexi of the gut, all right, and they are going to innervate, the motor function of these, the wall fibers of the hollow organs, and then you have also part of the nucleus ambiguus that has been for a long time considered just as a special visceral afferent for phonation and swallowing, but it also contains preganglionic parasympathetic fibers that reach the heart and serve as an input to the cardiac plexus here for the parasympathetic innervation of the heart. All right, that's the motor portion. Now, when you go to the general visceral afferent portion, which is essentially the sensory innervation of the viscera, the only nucleus in the nervous system, central nervous system, that receives the terminals of that is the nucleus of the solitary tract, all right? That receives now afferent fibers that have the unipolar neurons in the ganglion supporting them. They come into the nervous system and they terminate in the nucleus of the solitary tract. Very good. So we're making some progress. Now, an important consideration that you should keep in mind, the concept of plexi, all right? So you're familiar with spinal nerve plexi, the brachial plexus. What is it? Well, you have the rootlets coming off the spinal cord, and these rootlets are forming a plexus, which means that there is some kind of rearrangement of nerve trunks. But it's all nerve fibers. There is nothing else, right? It's just the rearrangement of nerve fibers ending up in particular nerve trunks, all right? Now, on the other hand, you have the concept of the autonomic plexus. There are plenty of autonomic plexi, as we're going to see later on, all right? And these are, again, rearrangement of nerve fibers, that they join and then they split up and they eventually go into more truncal systems, nerves, but has an additional feature, an autonomic plexus. In the crossings here, the corners, there are groups of neurons within the plexus. Here, there were no neurons here. There are nerve fibers. Here you have plexi, and these plexi have the arrangement of an autonomic ganglion. They are like that, so that you have an input that is cholinergic, and a nicotinic receptor on a projection neuron, and a muscarinic receptor on a CIF cell. And in this case, using the same example before, we have the dopaminergic neurons that now provide dopaminergic innervation through dopamine receptors in the projection neuron. So have this clear idea in mind. One thing is the spinal nerve plexi, the other is the autonomic plexus that contains little ganglia at the crossings. All right, now we have to go into discrete subjects. We are going to deal with the neural control of blood pressure, the neural control of urinary bladder function, and the neural control of sexual functions. Starting with the neural control of blood pressure, and perhaps we will we will stop after this for a break. Now, there is a long-term chronic control of blood pressure that is mainly hormonal, that we are not going to deal with it. It is not part of our subject. No? On the other hand, there are short-term, rapid, acute control of blood pressure that has to be through the nervous system. The humoral system is slow. It's slow to produce it, to reach the target, and so forth. This is an acute need of control of blood pressure that occurs, as I mentioned several times before, when from a reclining position, you suddenly, get up and stand on your two legs. There are so-called baroreceptors that can sample the blood pressure, right? And these baroreceptors are essentially, mostly there are several others, but let's say there are essentially two regions. One is the carotid sinus, which is at the bifurcation of the internal carotid into the internal carotid and the middle and the external carotid. At this level, there are baroreceptors in the walls of the carotid, and then in the aortic branch and the aortic arch. Now, the baroreceptors from the carotid plexus are innervated by the ninth nerve that has this nerve fiber, actual sensory fiber, as the usual unipolar neuron in the ganglion, corresponding ganglion of the ninth nerve. On the other hand, the receptors at the aortic arch are carried by fibers of the 10th nerve from the vagus, again with unipolar neuron supporting these fibers in the corresponding ganglion of the 10th nerve. Where are all these sensory fibers terminating? This you have to know already. These general visceral afferent fibers that we are dealing with, they go into the nervous system through the 9th and the 10th nerve, and they incorporate themselves into a system of descending fibers that is called the solitary tract, that has attached to it a nucleus. The nucleus of the solitary tract with these fibers terminate. Nucleus of the solitary tract is an extremely complex structure, receives input from several other regions of the brain, including the vestibular nuclei, and eventually whatever processing of information happens in the nucleus of the solitary tract, there will be outputs of the solitary tract. One output acts on the nucleus ambiguous and actually through various circuitries results in inhibition of the nucleus ambiguous. You remember that I just told you that nucleus ambiguous has the neurons of origin of the preganglionic parasympathetic control of the heart, and the action is slow in the heart. Now it's inhibited. The end result of this will be tachycardia, an increase in the heart rate. Now, the solitary tract nucleus also projects to the hypothalamus, as I mentioned before, and it stimulates the secretion of vasopressin. And vasopressin, one of the actions of vasopressin, is also vasoconstriction. Finally, and very importantly, the solitary tract is going to project to groups in the ventral lateral medulla, which in turn gives rise to descending fibers that are reaching the lateral horn of the spinal cord, where you have the neurons of origin of the preganglionic sympathetic fibers that, as you can see, are climbing the paravertebral ganglia and terminating in the ganglion, autonomic ganglia of the paravertebral chain. And stimulating that output will result again in an increase in the sympathetic tone, resulting in vasoconstriction. So through all these mechanisms, when these receptors are sensing a drop in blood pressure, as it occurred when you go from the reclining to the standing position, this thing will fire, and all these mechanisms will result in vasoconstriction and tachycardia, all mechanisms that increase blood pressure to the adequate level. All right. Now, dysfunction. What happens when this system fails? You have at least three syndromes that you have to understand. One is what is called orthostatic hypotension. All right? In orthostatic hypotension, you have a patient that indeed, when it goes from the reclining to a standing position, gets dizzy and probably faints also. All right? And this happens, it's a condition that has to be treated, of course, usually treated with increases in blood volume substances. I don't want to go into the therapeutics of that, but know that this exists per se as one syndrome, orthostatic hypotension. Then you have another thing that is called pure autonomic failure. Here, there is orthostatic hypotension, but is accompanied by several other features that reveal a failure in sympathetic control. All right? For instance, you have impotence, you have incontinence of urine, you have constipation, several symptoms that indicate that the autonomic system is at failure, together with the orthostatic hypotension. This is called pure autonomic failure. And then you have autonomic failure that is not pure, but is accompanied by other things in the central nervous system. This is what is called multiple system atrophy. And there are various forms of multiple system atrophy. One of them is what is called striatonigral degeneration, that gives you autonomic failure symptoms together with Parkinsonism. This is one a non-Parkinson disease Parkinsonism that doesn't respond well to L-DOPA, is different than Parkinson's disease, but has the feature of Parkinson's disease, the bradykinesia and the tremor and the rigidity, all right, in addition to the autonomic failure. That's one form of multiple system atrophy. Another form affects particularly the cerebellar system. That is what is called olivopontocerebellar atrophy. Olivopontocerebellar atrophy. The inferior olive and the pontine nuclei and the opposite cerebellum are atrophic and gives you cerebellar signs, ataxia, dysmetia, and so forth and so on, in addition to autonomic failure. Finally, and by the way, this also instead of cerebellar can be pyramidal, corticospinal, that gives you autonomic failure and a Babinski sign, the sign of upper motor neuron disease. And then you have an eponym, the Scheidegger syndrome, all right, that has features of nervous system, central nervous system dysfunction, but where autonomic dysfunction predominates. All right. At this point, we will stop, and we'll continue later or tomorrow. We'll see how the schedule goes with the neural control of urinary bladder and neural control of sexual functions. Thank you very much. All right, in the remaining time, I'd like to cover the neural control of bladder, urinary bladder, and of sexual function. We'll start with the bladder that, as you know, is like a bag, right, that collects urine from the urethers. And this bag can be empty at some point or semi-empty. And then you have to consider that in addition to the smooth muscle that makes the walls of the bladder, there is also around the urethra, in the posterior part of the urethra, a couple of striated muscle fibers, all right, that surround the urethra, and that when they contract, they act like a sphincter. But it's not really a sphincter, but in any case, we will represent it as such, something like that, all right? Something that squeezes the urethra and closes it, striated muscle. All right, in addition to the bladder, we have to see now the portion of the nervous system that has to do with bladder control. And don't get too concerned about this drama. What I'm trying to represent here are levels of the nervous system. For instance, this block represents a T10 to L2 level of the spinal cord, whereas this block represents an S2, S4 level. It's important because a lot of the bladder control that is carried out through the parasympathetic and the sympathetic nervous system needs cells of origin that are located in these regions of the spinal cord. As you know, the S2, S4 contains preganglionic parasympathetic fibers, whereas T10 to T2 maybe contains neurons of origin of the preganglionic sympathetic fibers. All right. Now, coming out of the spinal cord, you have, of course, spinal nerves that collect into plexi of spinal nerves. Remember the difference between the plexuses of the spinal nerves and the autonomic plexi that contain ganglia within them. So in any case, this may represent, let's say, the thoracolumbar plexus, right? And then from the lumbar plexus, you have various nerves that distribute in the corresponding territory, but in addition, they have a special nerve trunk that is called the splagnic nerve. That splagnic nerve actually contains the sympathetic fibers, and they are going now to go into sympathetic plexi, autonomic plexi, the concept of which I gave you already. All right, and they go from plexa to plexa. All these are located prevertebral in front of the spinal column. And so they have an initial one that is the celiac plexus, then a few branches go into another plexus that is like the aortic plexus, and keeps on going the same, and eventually gets to a superior mesenteric plexus. Now from that, now there is a nerve trunk that is of sizable lengths. These others are short, and they mesh with each other, one plexus with the next. But now comes a discrete nerve trunk that is called the hypogastric nerve. All right? And this hypogastric nerve comes down to the pelvis and actually opens up or terminates or develops into another autonomic plexus, which is the pelvic plexus. All right, that's about that for this portion of the system. Now, if we go now to the sacral cord, the sacral cord again has spinal nerves that go into a sacral plexus or lumbosacral plexus, and then there will be a special, from that plexus, there will be now a special nerve trunk that comes out of that plexus, and that is called the plexic nerves. It's also called the nervus erigens. And that nerve trunk comes into the same pelvic plexus that the hypogastric nerve was coming into. Now, in addition to this, the sacral plexus gives off now a peripheral nerve, somatic. And this peripheral nerve is called the pudendal nerve. So you see that there are a couple of things here. First of all, levels of the spinal cord that are critical. Secondly, all these plexi that end up here in a hypogastric nerve. Then you have the pelvic nerve or erigens that is important, and finally the pudendal nerve. You have like three nerve trunks that are going to control bladder function. So let's start with the actual roots. The impulses are taken to provide that kind of control. So you have here, let's say, in addition to this reminds me of another important level of control. All of this that I just described is also under control of upper central nervous system structures like the frontal lobes in the cerebrum, and also there are parts of the pons that exert descending control of these mechanisms. All right, now you have the following. I said that the sacral cord, S2, S4, contains preganglionic parasympathetic neurons. They are located at the base of the dorsal horn of that region of the spinal cord. And the fibers of these neurons, the axons of these neurons, take this root of the sacral plexus, they go into the pelvic nerve, and they come that way from the pelvic plexi. There are derivatives of that plexi that take care of the bladder and on the rectum and on the uterus and all the pelvic organs in general. The bladder is one of them. So these fibers are going to come into the wall of the bladder, and they are going to terminate in what are called intramural plexi. is within the wall, within the muscle layer of that wall, which is smooth muscle. There are terminations of these fibers, and they contact neurons that are within that wall, what is called the intramural plexi, right? And then a neuron there, a neuron at this level now has a short axon that is going to activate the smooth muscle of the bladder. What's going to happen? The bladder contracts. Now, it is important to realize that this kind of mechanism is essential for maintaining the same intra-bladder pressure within a certain volume that is accumulating. So urine is produced, from the kidneys to the urethra into the bladder, and that bladder gets filled more and more, all right? And so the pressure would increase in the bladder. You have here now another kind of spinal cord sacral neuron that is of purely somatic nature. These are alpha motor neurons and gamma motor neurons that they are familiar with the motor system in general, somatic, and they take the route of the pudendal nerve, and they are going to innervate the so-called external sphincter of the bladder to keep it essentially closed. Now, when the pressure within the bladder increases beyond a certain limit, what's going to happen is that there will be sensory endings in the wall of the bladder, and particularly at the bladder neck, right? That collects this information and sends afferent volleys again through the pelvic nerve, and they come to the spinal cord segment. And there, several things are going to happen. First of all, these terminals are going to provide excitatory influences on the preganglionic parasympathetic, and at the same time inhibitory influences on the motor neurons that innervate the sphincter. The end result of that is a purely reflex arc here that controls micturition, right? Because when you get a certain level of pressure and the afferent system comes into the spinal cord, by the way, this is a sensory fiber, right? It comes from the terminals here to the spinal cord here. But as you well know, the sensory fiber needs a cell body somehow to support it, right? And there is a dorsal root ganglion of that general level, which is represented by this cartoon. So you have here all the mechanisms to make an effective bladder control, right? When the bladder acquires certain volume and there is stretch of the wall here, these receptors, the sensory receptor, particularly the bladder neck, will send afferent impulses that are going to both excite the preganglionic parasympathetic, resulting in the contraction of the detrusor muscle of the bladder, and at the same time inhibition of the motor neurons that were maintaining the sphincter closed. Now, in addition to this local mechanism, there is also ascending pathways now that are being activated by the same sensory input, all right? They ascend the brainstem and the spinal cord and the brainstem, and they reach upper levels of the nervous system, to the pons and to the frontal lobes, to thalamus, so more complicated mechanisms, but schematically it reaches those levels, all right? These levels in turn are sending excitatory influences on that general circuit here. So this is a way in which now the upper nervous system controls these lower mechanisms. And it's well known, right, that at some point you feel the need to go to the bathroom, and however, it's not convenient for you, and you just don't go. And all this is defined by upper levels of the central nervous system. Now, when it passes this ascending pathway, when it passes through the thoracolumbar spinal cord, leaves there collaterals, right? And this, I'm missing one, there. And these collaterals now are giving rise to descending pathways that are of sympathetic nature. That means preganglionic sympathetic fibers that take all the route through the various plexi, and they may end in one of these plexi and is relatively undefined. It's not in a particular plexus that they end. I chose to make them end here in the pelvic plexus. It could just as well end in the superior mesenteric. But in any case, when it ends in that network of the autonomic circuits within the plexus, as you know, now there will be an output of that network. And that output, it will be such that it will come to the bladder wall, and that is going to control mostly the innervation of the bladder. They don't take much of a role in the actual contraction or relaxation of the muscle. Now, one more thing that I skipped before is that there are also inputs from the bladder wall, actually, sensory, that take the route of the sympathetic nerves, all right? But they are just sensory. And they are supported by unipolar neurons in the ganglion, as you can see here, and they terminate in this general region of the sympathetic networks of the thoracolumbar cord. What these afferents are doing is not very clear, but they may have to do with the sensation of fullness and particularly pain when there is distension or retention or whatever, and there is severe pain. These are the fibers that are carrying that particular input. All right. So these are the general routes through which the mechanisms are playing around. Now, you see essentially here that you have two critical nerve trunks. One is the pelvic nerve, and the other is the pudendal nerve, all right? The hypogastric nerve that carries these sympathetic influences apparently is not very drastic for the mechanism. But these two nerves that can be affected, by various reasons we'll see in a moment, are important to keep in mind for the control of the bladder. Now, let's go through the actual stages in the mechanism to control the bladder. First of all, there are what are called the intervoidal periods. To void is to urinate, right? So when you urinate, between this urination and the next, there is a period that you don't urinate, and that's the intervoidal period, all right? And now in this period, there is a stretch reflex that I already described in the mechanics before, that maintains a constant intra-bladder pressure in spite of the increasing volume, because urine keeps on being produced. All right. At some point, you feel the need to go to the bathroom, right? And now is the micturition reflex that I also described in the cartoon before. Above a certain volume threshold pressure, the pressure will increase, and that will result in afferent inputs, mainly from the stretched bladder neck, right, that will fire a volley of afferent information. And then this is going to activate the parasympathetic efferents that will induce contraction of the detrusor muscle. That's fine. At the same time, this reflex that is occurring all the time, right, is self-regenerating, and it fatigues very quickly, all right? And, it may last, when you want to go to the bathroom, it may last a couple of seconds that you feel the need, or maybe up to a minute that, I should go, but I cannot, I have to listen to the lecture or something of that sort, all right? And then the contraction of the detrusor muscle ceases, it stops. And then another reflex occurs when more pressure is built up. All right? So that means this reflex is self-regenerating, but fatigues after seconds or minutes, and the contraction ceases. All right. This reflex remains inhibited for minutes or maybe an hour, You can hold on for an hour and to go to the bathroom in the intersession or whatever, right? But the cycle starts again, and at some point, the contraction reaches a critical level. And at this level, there is inhibition of this nucleus that was providing somatic innervation to the sphincter, and now it's inhibited, and therefore the sphincter relaxes. And you pee, all right? Now, all this entire mechanism, as I said before, is under control to some extent of the frontal lobes and pons regions. All right, once you understand the mechanism of the normal function, let's go now a little bit about dysfunction, which is very important, all right? So I'm showing here exactly where we ended before when we were describing the various mechanisms in terms of neurons and fibers, right? Now, you can have problems up there in the cerebrum, frontal lobes, pons, all right? And that gives rise to what is called an uninhibited bladder. So you cannot really control it, all right? So these patients may be walking around and peeing at the same time. The bladder is completely uninhibited. Now, if you have now lesions, let's say, affecting the spinal cord above sacral levels, all right? And that can be at the thoracic level, at the cervical level, you have your spinal transections, whatever. The bladder becomes an automatic bladder, also called a spastic bladder. What does it mean? It means that reflexly the pressure builds up, and at some point the patient urinates automatically until the pressure again rises in a couple of hours or whatever, and he pees automatically. The problem that something is really missing here. All these descending influences from the upper levels of the spinal cord are not there anymore. And I don't know whether you notice that, but it's a matter of experience. When you pee, you end up your urination, and usually you contract somewhat voluntarily part of your abdominal muscles to actually relieve the rest that may remain from the automatic contraction of the bladder. So in this case, in the automatic bladder, that fails. And therefore, the patient automatically will do the urination, but there will remain always some residual urine in the bladder, and this is going to build up. And these patients require periodic catheterization of the bladder to remove that residual urine that is more prone to infections and so forth and so on. Now you have another lesion, let's say a lesion that involves either the sacral cord or the plexi or the pelvic nerves, the pelvic plexi, all right? It's all it's involving the whole mechanism. The whole reflex mechanism is destroyed. So what you're going to have? You're going to have what is called an atonic or flaccid bladder. What does it mean? It's a patient that accumulates urine to a certain extent and then leaks it out. All right? The bladder doesn't really contract. It's been outflowing just because of volume. Now, finally, you can have lesions of the peripheral nervous system. In the peripheral nervous system, you can get both these ending nerve trunks that come directly to the bladder, and also the pudendal nerve can be involved as a peripheral nerve. These are what is called the neuropathies, and one prominent neuropathy that gives this problem is diabetes, as a consequence of diabetes. So let's summarize that. These are important points that you definitely should retain, all right? You have a table here that I'll give you the bladder condition, the symptoms, and the localization of the pathology. So we discussed the uninhibited bladder. The symptoms are retention and in incontinence, and also urgency. You want to go to the bathroom very frequently and fast, all right? And ends up with incontinence. The pathology is in the upper levels of the nervous system, in the brain itself, meaning that you can have that in any kind of disease, like a stroke can give you that, demented patients do that, Parkinson's disease, and also multiple system atrophy will give this kind of uninhibited bladder. The next condition is what is called an automatic or spastic bladder that I discussed before. What are the symptoms? As I said, there is automatic emptying, but with residual, requiring catheterization. The pathology is in the spinal cord above the sacral level, right? The sacral level contains all the reflex necessary to do it, and as in the automatic bladder. But in this case, you have problems, a transection of the cord, you can get that in multiple sclerosis, anything that affects the spinal cord above sacral levels. Next, you have the atonic or flaccid bladder. The symptoms here is, again, retention and ending in incontinence, right? And those are lesions that destroy the sacral spinal cord or the plexi, all the roots, anything that is developing in the pelvis, like a tumor or surgery for that tumor or injuries will give you the atonic bladder, which is pretty bad. Finally, there is something that is called sensory bladder. In the sensory bladder, you have also these symptoms of retention that may end in incontinence, all right? And that's characteristic of some neuropathies, particularly of diabetes. Very good. That ends up the neural control of bladder, and let's go with some speed into the neural control of sexual function. Let's see. In sexual function, what do you have? Well, you know that, right? So you have stages in sexual function. The first stage, that is arousal and eventually erection. Then you have lubrication. Then you have emission. And finally, you have ejaculation and resolution, ending in resolution. So let's see how these mechanisms play around, all right? We'll do exactly the same layout as for the control of bladder. Being here at the thoracolumbar levels, being here the sacral levels, all right, the same as before. And here you have all the plexi coming off it, the sympathetic, the splenic nerve, and the various things, and eventually ends up in the hypogastric nerve that comes into the pelvic plexus. It's exactly the same, the layout. And in the sacral, from the sacral cord, now you have the pelvic nerve, or nerve erogens, is the nerve that provides erection, as we are going to see in a moment. And then you have the somatic nerve, which is the pudendal. All right? So it's exactly the same as for the bladder control. But of course, the mechanisms are a little different. First of all, you have here that everything at the spinal level is being controlled by upper levels of the nervous system, like the cortex and particularly the hypothalamus, because after all, this depends on an interaction between the endocrine system and the nervous system. You know, the endocrine provides hormones that act on nerve cells also, and eventually the hypothalamus will provide gonadotropins, for instance, which are essential for the development of the sexual organs. Anyway, let's go by stages, all right? First of all, I said that you can get arousal, as you know, from merely thinking about it, right? So that the upper levels of the nervous system are essential for this kind of so-called psychogenic eventual erection, right? And these pathways are going through, coming to the spinal cord, where they terminate in particular circuitry. All right. On the other hand, you have also arousal and consequent erection from stimulation of erogenous zones, right? Erogenous zones that are not only the genitalia themselves, also the perineum and even the lower urinary tract can give rise to erogenous sensations, all right? And so these sensory inputs, right? So these sensory inputs are coming through the pudendal nerve. So the pudendal nerve carries the results of local stimulation of erogenous zones, all right? And this is a sensory fiber, right? It needs, first of all, a neuron in the dorsal root ganglion to support it. And so here you have the confluence of psychic stimuli and regular sensory stimuli from erogenous zones. What do they do at the sacral cord level? Well, they activate here the preganglionic parasympathetic fibers that are going to take now the route of the pelvic nerve, and they are going to terminate in the pelvic plexus. And here there are things that occur, and the output of the pelvic plexus is going to go and influence erectile tissues. By the way, everything that I'm talking about applies the same to males and females with obvious differences, all right? So, but in this case, these parasympathetic, already postganglionic fibers here, what they do is they act by vasodilation. And this vasodilation is produced by a particular gas, which is nitric oxide, all right? And this nitric oxide results in the release of substances that dilate the arterioles and the trabecules in the erectile tissue, leading into engorgement of erectile tissue and actually erection, all right? And so this will be the first phase. So we dealt already with erection, and that's all that it happens, either from the central nervous system or at the local level. You may not need the central nervous system, as we're going to see later, all right? Now, this same parasympathetic output here, it also does a next thing, which is the contraction and activation of glandular tissue that gives secretions that provide lubrication. This, of course, this second stage of the sexual function is essential. Lubrication fails, forget about it, all right? Now, what provides the lubrication is mostly the responsibility of the female through two factors here. One is the secretions of the Bartholin glands, and the other is the transudate from the vaginal lining. Remember that the vaginal lining is not a mucosa, doesn't secrete mucus at all, and whatever it secretes is just a transudate, right? That's essentially female mostly. But in the male, there are also bulbourethral and urethral glands that provide additional lubricating material, right? Now, of course, these erogenous zones keep on acting. We're talking about, active intercourse. So more and more of these stimuli come in here, right? After the erection and the lubrication. And now these stimuli are activating circuits here in the same sacral spinal cord that go all the way up to the central nervous system through the spinal cord and brainstem. And at the thoracolumbar level, they leave some collateral. And that collateral is going to activate now a sympathetic preganglionic fiber that eventually ends in one of these plexi. I selected here the superior mesenteric, but it could be anyone. And then whatever happens in that network within the little ganglia that the superior mesenteric plexus has, there will be an output, and this output is going to take the route of the hypogastric nerve and then goes into the pelvic plexus and then eventually arrives to contract various things. Smooth muscle. First thing, it contracts the bladder neck. And that, of course, is important because it prevents the retrograde ejaculation. So it provides a closure of the bladder neck. And then it contracts these various elements that have smooth muscle the vas deferens that produces sperm, the prostate that produces prostatic fluid, the seminal vesicles that provide seminal fluid, and all this is, of course, in the male, but the female has also a small amount of prostatic tissue that is in the anterior, some spots of the anterior wall of the vagina, and these are called the skin glands, and that they correspond to what became more or less famous as the basis of the so-called G-spot. I imagine that everybody is familiar with that, but if you want to ask any questions, you're welcome to that. So in any case, what is happening here? There is an emission here of various substances, right? And they all converge and pour their effect into the posterior urethra. And that's what I'm trying to represent here. That's emission, and whatever is emitted goes into the posterior urethra. So the posterior urethra, on the one hand, is closed towards the bladder by the contraction here of the bladder neck. And so essentially it builds up pressure within the posterior urethra. More and more of these substances come into the posterior urethra. Actually, the mixture of all these three substances is what is called semen. All right? So the posterior urethra then gets with increased pressure and gives rise to afferent sensory information, right? And this sensory information travels also in the pudendal nerve. So the pudendal nerve becomes very important here. All right, and these sensory fibers are supported by unipolar neurons, and they reach the sacral cord. Now, in the sacral cord, these fibers are going to stimulate just motor neurons, regular motor neurons in the spinal cord, that are going to take the route of the pudendal nerve, and they are going to come into striated muscle and producing rhythmic contractions of the bulbocavernosus and the bulbospongiosus, and these rhythmic contractions eventually diffuse or expand into the perineal muscles and the pelvic muscles and even the trunk. All right, and all this is somatic, as you can see. But in the female, there are also at that stage rhythmic contractions of both the vagina and the uterus, and this must be sympathetic to some extent. So this part of this block should be taken, with a grain of salt, because it's not all somatic, but in the male is essentially somatic. All right. So that takes care of the fourth stage, which is ejaculation, right? And the fluid goes out. All right. Now, once that happens, there is a stage of so-called resolution. And in the resolution, various things can occur. Let me see if I can review that. The erection is parasympathetic, lubrication is parasympathetic, emission is sympathetic, ejaculation is somatic, and in the female there must be some sympathetic effect. And now know that the sum of emission and ejaculation constitutes orgasm. And finally, you have resolution, and the resolution, there is a reversal of processes so that the parasympathetic vasodilation goes down and the sympathetic vasoconstriction goes up. That deprives the erectile tissue with the engorgement that it needs. At the same time, the blood flow to erectile tissues decreases, right? Because there is vasoconstriction. And finally, the venous drainage that was more or less obstructed during erection gets completely relaxed, let's say, and there is venous drainage increased, and finally the resolution is completed. Now, important pieces of information are concerning the neurogenic sexual dysfunctions. All right, we went through the bladder, you remember, with the uninhibited, spastic and flaccid and sensory. Here we have to do it in some other, a little bit different, because we are going to put the location of the pathology first, and then we are going to see the results on psychogenic arousal and the result on reflexogenic arousal, just from manipulation, and then the effects on erection and on ejaculation. All right. Suppose the pathology is in the brain, right, up there. Again, example, stroke, dementia, Parkinson's, multiple system atrophy. So the psychogenic is very much impaired, right, because it takes origin in those areas. And the reflexogenic is also impaired. And the erection is also impaired, and so is the ejaculation. So you have, with this cerebral pathology, you have a problem throughout, not 100%, but very much affected the sexual function. Now, on the other hand, when you have a spinal cord lesion that is above lumbar levels, now, not sacral, above lumbar levels, what's going to happen? Well, if you have a transection of the cord, let's say at the mid-thoracic or cervical, you cannot have the psychogenic impulses coming down. They are interrupted, and therefore there will not be psychogenic arousal. Still, at the local level, is preserved reflexogenic, because remember, there was a very local circuit there to work without the need for upstairs influences. And reflexogenic arousal is preserved, and so is erection, of course, and so is ejaculation, which means that a person with a spinal cord level above lumbar levels, can have sex and can have kids. It may not feel much, but it can perform it. On the other hand, when you have problems of the cord between the thoracolumbar and the sacral, let's say L3 to S1, what's going to happen? Well, the cord is interrupted, psychogenic is gone, all right? Now, reflexogenic is all at the sacral level, and therefore will be preserved, right? And the erection will be preserved. But remember that for ejaculation, you needed the sympathetic that comes up to the lumbar cord and comes out from there and comes down then to the pelvic plexus. And that's destroyed here. And therefore, most probably, it will not be ejaculation. Finally, if you have a cord level at the sacral cord, or at the roots, or at the pelvic plexus, all right, you have the mechanism destroyed. And therefore, you have zero throughout. That's it, finished. Finally, if you have a problem with the peripheral nerves, like in diabetes, for instance, what's going to happen? What's going to happen is that psychogenic will be impaired, and reflexogenic erection will be quite impaired, practically, yeah, very impaired. And then, the reflexogenic arousal is impaired. So the arousal, both psychogenic and reflexogenic, are impaired. Now, in terms of erection, in peripheral neuropathy like diabetes, is the typical example of so-called erectile dysfunction, means total impotence, all right? And finally, ejaculation could be possible. You don't need erection to have ejaculation. All right. All right, this finishes the neural control of sexual function. So a couple of take-home messages from the entire autonomic nervous system that we were discussing for the last 2 hours. All right. First of all, remember that visceral sensory fibers, GVA, terminate in spinal cord for the pelvic organs and the solitary tract nucleus for thoracoabdominal viscera. Now that's clear. Now, visceral motor fibers, GVE fibers, all right, can be preganglionic parasympathetic that originate in brainstem nuclei and sacral cord, or can be preganglionic sympathetic originating in the thoracolumbar cord. Both of these preganglionic fibers terminate in autonomic ganglia. Remember that autonomic ganglia contain networks, not simply pre-post synapses. And finally, the autonomic plexi contain small ganglia at the crossings. which makes them very different than the regular spinal plexus. All right, that's in terms of the general autonomic nervous system, various points. Now we went through the acute control of blood pressure, and we said that there are baroreceptive fibers that terminate in the nucleus of the solitary tract, and the solitary tract output ends up inhibiting the nucleus ambiguous and resulting therefore in tachycardia. They also excite thoracolumbar lateral horn preganglionic sympathetics, resulting in vasoconstriction. And they also excite the hypothalamus, that results in a surge of vasopressin that also leads to vasoconstriction. So all these mechanisms tend to increase the blood pressure. Now, failure of this mechanism, you get orthostatic hypotension, you get pure autonomic failure, and you have multiple system atrophies where the autonomic problems get together with either Parkinsonian features or corticospinal problems or cerebellar problems. Now the last thing will be the review of the bladder control. You have an intervoiding period, stretch reflect, increased volume with no increase in pressure. The micturition reflects above a certain volume. The afferent volley through pelvic nerves goes to the sacral cord. And then excited parasympathetic efferents to intramural plexi of the bladder eventually end up in contraction of the detrusor muscle. It is the inhibition of onus nucleus, which is this motor nucleus in the sacral cord, leads to relaxation of the external sphincter. And the entire mechanism is under central nervous system control from frontal lobes, from the pons. Finally, the neurogenic voiding dysfunctions. You have the uninhibited bladder, urgency, frequency, incontinence from cerebral lesions. You have the automatic bladder, is automatic emptying with residual in spinal cord lesions that are above the sacral levels. Then you have the atonic bladder, that is retention leading to incontinence that occurs in lesions of the sacral cord or roots or pelvic plexus of various pathologies. And finally, the sensory bladder, that is also retention or incontinence. This is problematic, that occurs in neuropathies like diabetes. And finally, the neural control of sexual function, where you have the arousal, stimulation of erogenous zones, depends on integrity of the pudendal nerves. That's important. The pudendal nerves are important for that, right? From the diagram, the cartoon that I showed you. Erection and lubrication is parasympathetic and depends on integrity of the pelvic nerves. This is pudendal, this is pelvic. Then emission is sympathetic, depends on integrity of the hypogastric nerve that was descending from the superior mesenteric into the pelvic plexus. Finally, ejaculation is somatic in the male and in part sympathetic in the female, depends on the integrity of pudendal nerves again, and in part hypogastric nerves too, for the sympathetic portion. Finally, the resolution is mainly sympathetic, is occlusion of the parasympathetic and increase of the sympathetic flow. Now, in terms of dysfunction, this is the function. In terms of dysfunction, you have that the function is totally abolished in lesions of sacral cord, roots or pelvic plexus. The reflexogenic ejection and ejaculation are preserved in spinal cord lesions above lumbar levels. And finally, the reflexogenic erection, but not ejaculation, is possible with lesions between L3 and S1. So that's all, folks. Thank you very much.